All right, everyone. We're going to go ahead and start our session. It's good to see such a nice group and that we're all in here to talk about taking action. So first, I'm going to give a quick recruitment and retention committee update. I'm Allison Caban Holt. I am the chair of the recruitment and retention committee. I was asked by my colleague, Tamara Mertz Rivera, to tell you a little bit about what I do in my day job. So I'm at the Wake Forest School of Medicine in North Carolina, and I am the associate director of the Maya Angelou Center for Health Equity there. And so I work regarding disparities across different diseases, including Alzheimer's disease, which is a focus for us. Many of the issues that we run into in Alzheimer's in terms of different populations that have been traditionally underrepresented in recruitment are very similar to what we're facing in cancer. So I think by understanding some of the principles and really putting together some best practices on how to do diverse recruitment and looking at the populations who are most affected by these conditions across diseases is going to be a great way for us to really move the mark and to really, move forward in terms of our diversity, equity, and inclusion. So I'm only going to give a quick RRC update. Our mission reflects one of SWOG's guiding principles, having diversity in membership and leadership, and as participants in clinical trials. And we are here to help advise research teams on effective methods for recruiting and retaining diverse groups of patients onto SWOG trials. I wanted to introduce Jamie Sundstrom, who is here. She is our new recruitment specialist. She is replacing Jennifer Mazers, that many of you may know. And Jamie has been great. She's going to be responsible for working with study teams in meeting their accrual goals by advising, developing, and implementing strategies to enhance participant enrollment. And we have some new support resources. There's actually now a recruitment and retention assistance request form. So in the past, teams may not have been aware that the RRC was available to them as a support and to talk about various issues that they may be having with accrual. We really are encouraging teams to reach out to us at an early stage, hopefully even before you've completed everything on the protocol, but reach out to us so that we can see if there are ways that we can advise you in terms of how to have effective recruitment of your patient populations that you are looking for. The RRC has members that have a number of different areas of expertise that you can avail yourself of. So please reach out to us. So we have this recruitment and retention assistance request form. And so I'm going to show you a little bit of the form. As you can see, there's not a lot of information that's requested, and Jamie will get back to you and we'll see if you can actually have you join us for one of the RRC meetings and present the study for your team. So please reach out to us. And we also wanted to share a few slides from the Hope Foundation, and there are a number of pilot grant awards. So you can see that there is one that's $50,000 for 1 to 2 years, and those applications are due June 1, so that's only a few weeks away. Early exploration and development seed funds, also $50,000 awards that are due July 1, and I think that should be July 1, 2023. An impact program award, that would be a $250,000 up to $250,000 awards for innovative SWOG research leveraging resources from trials. And some of the other deadlines have been extended, such as the Coltman Fellowship and Career Engagement Awards. Both of those are due September 1. And Hope Foundation is celebrating 30 years. And they have a Share Our Story, so you can share messages through their toolkit. And the Hope Foundation was founded 30 years ago, a full generation. And to honor this and further Hope's impact, we're raising $300,000 to fund the next generation. We need your help. So anyone who wants to donate to the Hope Foundation, please take a picture of the QR code. We know that the research that we do, and we know how Hope supports this research. So we know that the money goes to good use. And lastly, there is a call for Hope board members, and they are inviting nominations for board members this year. And so if you are someone, you or someone you know could be a good candidate, they have a short application that you can access at this link. Or take a picture of the QR code. And the goal is to promote strength and inclusivity in the nomination process. So they're encouraging all interested candidates to apply. So if you or someone you know would be a good board member for the Hope Foundation, please feel free to nominate. All right. Now we're going to move forward with the Take Action Symposium that was developed by Kamara Mertz Rivera and Lucy Gansauer, who are the Education Subcommittee co-chairs.
Hello, everyone. I'm Kamara Mertz Rivera, and as Allison said, I am the Subcommittee Education Chair for the RRC. And what I do in my day job is I am the administrator for Upstate Carolina INCOR, which is located in upstate South Carolina. And I'm the director of clinical research for our cancer center, Gibbs Cancer Center and Research Institute in Spartanburg, South Carolina. So today we are having our sixth, or rather fifth, Take Action series. It started back in 2021, and Take Action series was kind of a brainstorming session between Lucy and I and Allison and the RRC team to coin something that literally, this is why it's called Take Action. We have been talking a lot about diversity and the need for expanding enrollment to different populations. So our Take Action is our coin, and I'm really happy that we are here today to talk about reducing structural barriers and ensuring equity in clinical research. And I just took it upon myself to Google what a structural barrier is, and I thought it was a perfect opportunity for me to share from Google what that came back, and I think it's perfect. Structural and system barriers are obstacles that collectively affect a group disproportionately and perpetuate or maintain stark disparities in outcomes. Structural barriers can be policies, practices, and other norms that favor an advantaged group while systematically disadvantaging a marginalized group. So with that being said, I'm looking forward to having our presenters. And prior to that, I just wanted to ask everyone, there should be a yellow sheet going around that we are asking you to sign in. And there is a feedback form that I was told is in a salmon color. I see them out there. We'd really appreciate that if you could complete those. It's real simple, but some feedback about what you would like to see maybe in the next opportunity that we have for education in October. And lastly, we have virtual attendees as well. So we ask, we're going to have a Q&A session and invite additional panelists up towards the end. And we ask if you're going to ask questions, please go ahead to the microphone because I'm sure they'd be very interested in hearing what you have to say. So this is just an outline of what we're going to be going over, and we're going to start by reviewing some trial design and novel approaches, followed by a discussion of patient travel reimbursements and SWOG lessons and implications, and ending with trial design and improving representativeness. And then we'll have our Q&A. So I'd like to, these are our speakers. Today we have two that are in person, and that is Dr. Regkamp and Rick Bangs, followed by Dr. Shabra. Couldn't be here, he's here virtually, and he has done a presentation, a great presentation that we'll be playing for you at the end. So I'm going to start out by introducing Dr. Regkamp. She is a professor in medicine and director of Division of Medical Oncology at Cedars-Sinai Medical Center, and associate director of the clinical research for the Samuel Ocean Comprehensive Cancer Institute. She's received her master's degree in clinical investigation from the Department of Biomathematics at UCLA. And her research focuses in the development of novel therapies and biomarkers for lung cancer with an emphasis on targeted therapies. She is involved with the National Comprehensive Cancer Network Guidelines Committee from 2008 to 2020, providing guidelines on thoracic malignancies. And she's an active member of SWOG and the study chair for SWOG 2302 Pragmatica Lung Trial. So we're going to go ahead and forward the slides. There you go.
Thank you. So I appreciate the opportunity to speak today to this group, and you'll be hearing a lot about this trial and I think this design throughout the SWOG meeting. And so this will be, if you miss anything, you'll hear it again, most likely. And I'd like to thank Lucy Ganzauer for inviting me today and being very proactive as we develop this study. So I wanted to talk, so this is specifically about S2302 and Pragmatica Lung, which opened in the beginning of March. But really it's about how do we think about pragmatic trial designs and trying to increase inclusivity and reduce burden on our sites to get more trials open to more patients. So it's really about patient access at the end of the day. So the background of this trial, this is the trial, this is the schema. It is very simple. It's patients who have basically refractory non-small cell lung cancer who have received prior immunotherapy and chemotherapy, and they're randomized to get standard of care versus the experimental arm, ramucirumab and pembrolizumab. And where we have 700 patients that we're going to randomize, and the standard of care is really what the investigator would normally treat their patient with. And you'll see a little bit more. So this is really empowering our investigators to treat patients how they normally would. So what we know is that this is a population of patients. So why was a pragmatic design something we could do in this population? And so first off, this is a population of patients where the standard of care is still docetaxel and docetaxel ramucirumab, and these have been approved for over a decade. Response rates are limited, less than 30%, and survival is limited with those options. So we've been trying to move that needle. We've done a great job in the frontline setting, and most patients do receive immunotherapy and have benefits from immunotherapy, but there are still many, many patients who develop progression and don't have long-term benefits from immunotherapy. So the S1800A trial was performed under the LungMap protocol and was a phase 2 randomized trial of ramucirumab and pembrolizumab versus standard of care. So very similar type of design. And this trial it showed that there was an improvement in overall survival, 14.5 months versus 11.6 months, with a hazard ratio of 0.69, which is pretty substantial and impressive. There was 130 patients enrolled in this trial and randomized in this trial. And so it was a smaller study, phase 2. So the results are impressive. Most people felt that it needed more data in order to be put into practice. So then with a lot of discussions with the SWOG, with CTEP, with the FDA, we discussed the fact that we really just want to know survival. So could we put a trial together that just looked at survival. And then the other important piece of this is that we know a lot about ramucirumab and pembrolizumab. They've been treated, thousands of patients have been treated with both drugs, combinations of these drugs. And so we know about the toxicities, we know about the safety. And in our phase 2 trial, the safety of the combination of ramucirumab and pembrolizumab was better than what we see with standard of care chemotherapy. So safety was not a big concern. We want to look at survival. So this is really a trial that is asking one specific question, and we tried to get rid of all of the noise that we collect and burden our staff and our patients with, other than just looking at survival. And so the clinical trial design is supposed to reduce the clinical burden of trial participation on the patient and promote the inclusion of patients with this disease, in addition to decreasing the burden on the staff. And this is just the background. So this is the overall survival from our phase 2 trial. And again, you see clearly that there's a benefit for ramucirumab and pembrolizumab. Most of the patients in the standard of care received the kind of the highest, the best treatment that we have, docetaxel and ramucirumab. And again, we have a hazard ratio of 0.69. Most patient subgroups did benefit from this. And so we're including a similar population with a little bit broader eligibility in this phase 3 trial. And the progression-free survival. So importantly also, the progression-free survival was not, the median was not different. You can see that there's a tail that is improved with the ramucirumab and pembrolizumab, but the progression-free survival was not different between the two groups. And so that helped us to understand that maybe we don't need to collect even progression-free survival. And so imagine a trial without RECIST reads. Anybody who has to follow RECIST reads and amend those reports, it's a bear of a thing to do. So this is the toxicity, and again, what we saw were that overall that the toxicity was higher in the chemotherapy, the standard of care arm, compared to the ramucirumab and pembrolizumab arm. So the goals of this is to, number one, empower our investigators to really treat patients how they would in the real world practice. This is a little different than a real world design where they generally are looking retrospectively at how patients were actually treated. This is allowing them prospectively to treat patients as they normally would. And again, we'll look at the eligibility to talk about barriers to enrollment and significantly decrease data collection. Every site that I've encountered has had issues with staff retention and getting data in in the post-COVID era. And so minimizing data collection, improving that burden on the sites and the staff. And again, to the pragmatic design, making decisions on effectiveness, comparing the balances and the burdens of risks. And we really just want to look, is the efficacy there? Yes or no? Does this work under the standard conditions that we normally treat our patients? This is a registrational trial, which I think is unique in oncology to have a pragmatic trial in oncology, but I think it's not only registrational trials that could be pragmatic, it's really about trying to get the bare bones of what you actually need, answering a question and collecting what you need to answer that question. So our primary objective is to compare overall survival between the two arms. Very simple. And then secondarily, again, as we talked about, we know the toxicities of these drugs. So rather than every toxicity and putting those in and every start and stop date, we are only collecting high grade, greater than grade 3 treatment related adverse events and unexpected. So, so we will not be able to report on all of the immune related AEs in this study because those are generally expected and would not necessarily be reported. So here's a very busy slide, but the reason we put it up this way is because this is the eligibility. There are no other eligibility for this study. It is this short. And it's really about patients having metastatic or recurrent disease, having received PD-1, PD-L1 therapy, having received chemotherapy with progression, and allowing patients who may have received these in the adjuvant or neoadjuvant setting, if they've progressed within a year, that also counts as their therapy. We are allowing patients with sensitizing mutations as long as they've received the standard therapy and also meet the criteria of having received PD-1, PD-L1, and chemotherapy. The other piece of this is we're looking at acquired resistance. rather than primary resistant patients. So they have, in this study, they have to have had at least stable disease as their best response for at least 84 days on therapy. And we are including patients with a performance status 0 to 2. So I think that's important too. This is one of the, I don't, most studies using immunotherapy have not used performance status 2. So again, stage 4 recurrent, prior treatments, performance status, and safety. Our treatment A is standard of care. And we also acknowledge that standard of care may change during this 700 patient trial. And there are lots of trials that are out there that are phase 3. So we are really allowing investigators to choose whatever they would normally choose for this population. So we guide them to the NCCN guidelines, but they should be using what they would normally use in their practice. And they should use these drugs as they would in their practice. And so we don't put a lot of stipulations into how they should do this. We don't have to collect infusion start and stop times. We don't have to collect what medications they're getting with the infusions. They're getting it as they would in their standard practice. And the ramucirumab pembrolizumab is given at standard doses on a 21-day cycle. We're not allowing the 6-week cycle for pembrolizumab just to keep it uniform in the group. And these drugs are supplied for the study. And so here is our study calendar. And again, similar to the eligibility, this is it. This is the study calendar. And I think one of the remarkable things as sites are starting to put this study into their system and build beacon builds and things like that, we get lots of calls saying, where are the labs? Where are the scans? When do we do these things? And it really is, again, how the institution and the investigator normally practices. That's how we want you to practice. So there are no labs to determine eligibility. If you would treat your patient based on what their labs are, they would be allowed in the trial. And so it is based on the investigator's judgment for that. So we're looking, and then so overall, we're looking at vital status assessment. So we're always collecting survival, the SAE assessments, and then again, the provided ramucirumab and pembrolizumab. And one thing that is missing from here too is imaging. So imaging are done as per institutional guidelines. We don't specifically collect the RECIST for progression. And so we ask at each assessment and each treatment whether the patient has progressed, but we don't collect that specifically. So we have a significant decrease in data reporting. And I'll just go, so here, these are the number of scheduled case report forms that are required. And usually this is pages and pages, but this is it for this. And the vital status form is the most important form. There are a number of eligibility criteria and upfront demographics. We do collect genomic data for the patients and PD-L1 status upfront, but then there are treatment summaries and vital status and AE forms, and then finally the notice of death. These are just samples of these forms, just to show you how relatively simple they are. And again, this was a lot of collaboration and discussion. And one of the conversations that came from CTEP when we were putting this together was, if this doesn't make you uncomfortable, you're not doing it right. And so this is supposed to make us uncomfortable. And so when we see this, we constantly look at this and say, should we be collecting more? Should we do more? But it was really about doing less and only collecting what we absolutely need. And again, it's the high grade AEs that we're collecting. Treatment study, so treatment summary forms, what did the patient end up receiving? And again, we allow for almost anything. We put the standards generally there, but we allow for anything that an investigator would treat their patient with. And then the notice of death form. We ask for the pathology report to be uploaded. We want the PD-L1 and NGS to be uploaded, but we do not collect tissue specifically for this study. Most patients with non-small cell lung cancer will have some genomic testing and PD-L1 assessment. We do not collect imaging, but we ask the investigator whether the patient has progressed. Informed consent can be remote, and the informed consent has been greatly simplified. it's even shorter than the standard NCTN templates. This has additional funding as it's a registrational trial, but interestingly, it actually has less monitoring and auditing than a standard registrational trial because of the limited data that is collected. And so we are also trying to enhance our recruitment. So with the help of this committee and Frank in our I don't know, what is his title? Frank's title, I can't think. Huh? He takes care of everything. But working to get the recruitment efforts out there. We're working with a firm to evaluate our recruitment over time, so we'll be able to assess this. We have materials that are going to be in Spanish and maybe translated into other languages. We've been working with our advocacy organizations, and we'll be developing other materials as needed for support and awareness. We have monthly site support calls, and then there will be quarterly newsletters to recognize high accrual. And we had accrual within 2 weeks. We had multiple patients already accrued to the trial, and it's a number that keeps changing every day, but we're over 15, I think, right now. So, and then there's enhanced outreach to support sites with high numbers of patients with underrepresented groups. And helping to develop community engagement. We have a community engagement from the lung committee liaison, and we're continuing to monitor the representativeness of our enrolled populations throughout the trial. There are possible targeted funds for community sites, and some of those things are continuing to be worked on. We're also working on patient reimbursement. And again, we don't want to increase the burden on the staff. So these are generally in the form of gift cards that can be utilized easily. And again, this is something that SWOG is looking at overall for where this fits well. But this is something that we are moving forward with this study. We have resources in development, patient-friendly plain language trial summaries that are being worked on, and there are a number of training items in the CTSU site. Again, for ease of implementation and activation. So I think that's the main topic. And again, we're excited about pushing this forward and having really a reduced burden trial for both patients and study sites. We've already seen a large number of sites activating, and sites in the community sites, or more remote sites, excited to see that they could treat as they do in normal practice. So the barriers to needing extra labs, extra scans, pulling in scans from other areas, none of those things are required. So, and then it all takes a village. And so we have support from Merck and Lilly. We have support from all of our NCTN partners. And my co-chair is Konstantin Dragnev, and then FNIH and Friends of Cancer Research helped to spearhead this along. Our ops team, and Mary Redmond, who's our lead statistician, and her team, our data coordinators, and everybody on the team, our project manager, Mariah Norman, who helped to move this along at a very rapid pace. So thanks to everyone, and thank you for your attention.
Thank you so much. I'm very excited about this trial. We're trying to activate it now at my site, so I'll have to report back. Next, we're going to invite Rick Bangs to come up, and he is a bladder and prostate cancer survivor and serves as chair of the SWOG Patient Advocate Committee, SWOG Bladder Cancer Patient Advocate, and the Bladder and Penile Cancer Patient Advocate for the National Comprehensive Cancer Network, and is one of the NCI Cancer Care Delivery Research Scientific Steering Committee Patient Advocates. He co-chaired the NCI CCDR Intervention Breakout Group for a November 2022 clinical trials planning meeting, addressing health-related social needs to improve cancer care delivery and outcomes among newly diagnosed cancer patients in community settings, and a NCI Council of Research Advocates Working Group on clinical trials enrollment and retention focused on the intersection of accrual disparities and financial toxicity. He's also one of our RRC members, so welcome. And we look forward to talking about travel reimbursement. And again, we'll have Q&A at the end of this session, so stay tuned. Thank you.
So I want to thank Kamara and Lucy and Allison for inviting me today. This is my last meeting as the bladder cancer patient advocate. We have term limits that I instituted in the patient advocate committee. So I just want to introduce Daryl Nakagawa, who's in the audience. He is my replacement. So please introduce yourself to him. I found this quote from Albert Einstein. I think it's appropriate. In theory, theory and practice are the same. In practice, they are not. In theory, I should be presenting results at this meeting. In practice, I will not. Okay, so this is what I want to talk about. Some considerations for supporting participants. There are some guidelines out there that can be leveraged if you're trying to make a case for this, and some challenges and opportunities based on several years' experience coming down this path. All right, so from a GU perspective and a SWOG perspective, in April 2017, we kicked off a bladder trial. I was asked to present patient considerations, and then there are two that the vice chair, the late vice chair of the GU committee, honed in on, which was logistical burden and financial toxicity. And he asked the question, well, why aren't we doing something to reimburse patients? All right, so that was the genesis for this particular project, but note that the date was April 2017, and we are a few years later. All right, so from there, we went to the Hope Foundation. They do wonderful work, as everybody knows, and they were willing to fund a project. And so the first question we had was, well, how are we going to do this? What are we going to do? Is this going to be a randomized clinical trial? Because somebody's going to want to know whether this is effective, et cetera. And so while we were churning on this, Genentech had another trial. So the trial that we just talked about was a Genentech trial. But they had another trial coming down the pike, which had a little bit more of a travel issue, all right, because there was a surgery, chemo, radiation, and immunotherapy. All four of those were part of this. So then I was put into the amendment queue. I don't know how many people have been in the amendment queue, but it does not free up very quickly. And so several years later, here I am. All right, so there was mention of this working group that I was on. This is information that's available publicly, but there was a conclusion which wasn't surprising to some of us who deal in this business, which is that the interventions targeting reducing and overcoming financial costs have not been adequately studied. This is an understatement. All right, so then there was this clinical trials planning meeting like 2 years later, and it was specifically on health-related social risks or social determinants of health. Transportation was one of the 5 domains, along with food insecurity, utilities, housing, and interpersonal safety. Okay, not surprising for some of you in the audience, but apparently surprising to some others, was the fact that there was a lack of robust evidence. So we should not look at this as a bad thing. It's an opportunity for every one of us in this room, so we can actually mobilize and fill some gaps. All right, so the first lesson that I'm going to give you is there is a lack of evidence, and it's an opportunity. All right, the second lesson has to do with quantification. All right, so I want to walk you through the math for this particular trial that we're talking about, because people tend to relate better if you start looking at it holistically. So we're talking about 35 treatments. This is in the arm that does not include immunotherapy, which would add another 7 treatments. If you use an average of 25 miles each way for treatment, and I dare you to try to find somebody who will tell you what the average distance traveled by a patient on a clinical trial is, but we use 25 miles. We use the IRS cost at the time. Since COVID, the costs have gone up, but we use the cost of the time that was 57.5 cents. And parking is not cheap. Those of you who have parked at a cancer center know that that's typically not the case, although some are an exception. So that leads to $55.25 per visit. All right, so if I go in for a treatment or a scan, that's what it's going to cost me. So it was $1,500, and people start relating when you tell them there's $1,500, and you remind them that people don't have $500 or $1,000 sitting around for an emergency expense. That's typically the kind of number that's quoted. All right, I've excluded surveillance and other follow-up. So it was a pretty hefty bill. Under the new IRS guidelines, it would be another $200. Wrong way, sorry. All right, so now let's talk about the guidance that's out there. So the lesson to be learned here is language matters. All right, so you're going to see three terms I put in red: remuneration, compensation, reimbursement. Using the right language makes a huge difference in getting this approved. All right, so we need to talk about the right kinds of things here. And reimbursements are usually okay. All right, so the guidance says reimbursement is okay. And I'm differentiating that with getting some money to participate in the trial. The FDA guidance is similar. You'll notice here they talk about incentives and payment for participation, and they make some differentiations there. But the key point here is that the FDA does not consider reimbursement for travel expenses to and from the clinical trial site and associated costs as undue influence. All right. So this was important here at SWOG, because when I had to run this through the executive committee, the first thought that came to their mind was coercion. All right, so this helped counteract the coercion thoughts that people were having. All right, so we set up what seemed to be some rather simple objectives for our 1806, our bladder cancer trial. All right, so think of this as a proof of principle or a proof of concept. So I want to know who the stakeholders are. When we started this process, we didn't know how many people were going to touch this and what roles they were playing. We wanted to understand the steps to get through this and the decision points. We wanted to know something about indirect costs, which I learned a lot on this project about indirect costs. So some of you in the room may know what indirect costs are, but the sites take a cut, or SWOG takes a cut to cover overhead. Right. So as a business person, I understand the concept, but when I heard some of the percentages that are used for indirect costs, they were significantly more than I as a business person expected. All right, we want to understand how the sites are going to account for these indirect costs. In other words, were they going to absorb them, or were they going to take a cut from what you were giving for them to reimburse the patients? We wanted to understand the rate at which patients and sites cannot or will not participate. We went in with no illusions that every site or every patient was going to participate. We wanted to find out how many would. All right. And we wanted to understand how to get through the NCI approval process. These are the key stakeholders that we have uncovered at this point. All right, so you can see obviously patients have to be considered. We have to have somebody has to fund this, right? If we're going to do this, somebody's going to have to fund it. In this case, we had Genentech funding, but if this was a surgery trial, who's going to fund it? That's a question that has to be resolved. All right, within SWOG, we had the principal investigator, the patient advocate, which was myself, ops, contracts, stats. The EAC had to approve it. It has to run through the clinical trials partnerships team and some of the administrative work that happens there. And it needs to be supported by committee leadership. In our case, the Hope Foundation played a role. And then in order to get this approved, we have to get it through IRBs, whether it's central IRB or site IRBs or a combination. And then at the site level, there's administration folks, which might include accounts payable, right? So if you're going to reimburse patients in some contexts, if you're not giving out gift cards, you may have to go through accounts payable to get a patient reimbursed. Okay, I'm not gonna drain this slide, but an understanding of the process, we're much clearer on what the process we proposed was going to be. But if you're going to do this, you have to kind of understand how money starts from the funder and gets to patients. So you really need to understand each of the steps in this process. If you're going to do this, there's going to be some desire, rightfully so, to assess what you're doing. All right. So we set up a simple survey for the patients. At least we thought it was simple. We set up a simple survey for the sites because, again, this is proof of concept, but we wanted to know a little bit about how we could do this better. So that was part of our lessons. All right. And so I've included the survey in case people were interested. It can be used maybe as a starting point for other folks. Okay, so this is hot off the press. So 2 weeks ago, we got a disapproval from the NCI. All right, so if we step back and we think about how to move forward from this, what does this disapproval tell us? So the first comment that struck us was it says the NCTN program, remember this is a treatment trial, so it was flowing through the NCTN. So the disapproval says that the NCTN program does not support this type of research question and that financial matters are not, meaning should not, be discussed in the NCTN protocol document. This leads to a number of questions. You can see some of those questions. We have taken our folks at the NCI at their word and asked to have a discussion with them in preparation for the next round so that we know how to get through this process and what the right pathway is, what the right documentation is, where does this documentation belong if it doesn't belong in the protocol, and also who the approver should be. Okay, so this was the offer that we received that we've taken them up on. That meeting is scheduled for next week. All right, so we want to understand the comments that are made here. There was a comment that this was not going, in their opinion, this was not feasible. Again, it was a proof of principle, and we were not expecting every patient to be paid or every site to participate. So, and we were well down the road. By the way, this trial's been open for several years. So remember I told you we were an amendment, right? So we are well down the road. And frankly, my goal was to pay one patient. That's all I wanted to do, because then I could say end to end, we understand how to get this done once, maybe not the second time, but we at least understand the first time. All right. So there's a lesson here around pre-approval and making sure we understand in advance. You may want to have a conversation with the NCI about what you're going to do so you can figure out the right pathway, the right people. Okay, so our questions around the feasibility are, did they not think it was feasible to pay a patient? Did they not feel it was going to be feasible to execute the surveys? Did they feel like no sites would participate, or maybe they thought all sites should? We were not intending all sites, but maybe that's what they thought. All right, so there's a lesson here around expectations. Okay, there's a similar disapproval for a SWOG concept. I won't go through it in detail, but there's a lot of parallels here. All right, so I think this is an NCTN issue. This is not, SWOG didn't know what it was doing. Some of our counterparts are trying to figure this out as well. And so we've got to make sure that the right mechanisms are used here. All right, you'll note that DCP in this case declined to actually evaluate it. So I'm not sure who's supposed to evaluate these things, but we'll figure it out. There is some other work that Dr. Shankaran is doing that may be helpful in this regard. I only found out about this a day before the slide deadline, so I don't know anything more than just the title of the work she's doing, but it may be helpful for us as we move forward. All right, so these are some of the fundamental questions, and they fall into three categories: who, what, and when. All right, so who's going to fund it? That's a universal question. Doesn't matter how we're doing this. Somebody's going to need to fund it. Who's that going to be? Who's actually going to be paying the patients? All right, so in the Pragmatica trial, we have gift cards, which circumvents a lot of the issues that we were having here, and that's a good thing. All right, but somehow you got to figure out how does that get into the patient's hand. All right, who's going to qualify for payments? Is it going to be a universal truth, or is there some kind of screening and eligibility and some threshold that has to be met and so forth? All right, what amount are you going to reimburse for? And are you going to split this, or is it a one-time payment? Does it vary by distance or demonstrated need or receipts, or can different institutions do different things because the indirect costs are different? What kind of documentation? Remember I said that the accounts payable departments we knew were going to be involved. The way the accounts payable departments that some of the institutions work is they have the patient fill out a W-9. All right, that's an IRS document. IRS reporting is not required under $600, but even if you're giving them a $500 patient, that's the process that they use. Some patients are not really thrilled about filling out IRS documents on something that's theoretically not being reported. All right, so that there's some issues there. What is the form of payment? Gift card? Is it cash? Whatever. What controls are in place? Somebody's going to want to manage gift cards or manage cash. We don't want things to be just kind of running out of the cash register. And then the IDCs are certainly important issues. All right, I put under the when, I put this issue of retro payment. So we were putting it in as an amendment. Genentech wanted us to pay all patients. Okay, so the only way I can pay all patients is to retroactively pay people who are already participating. All right, so this was kind of going to be, for us, this was going to be a struggle because we weren't sure how the sites were going to execute a payment for somebody who was signing up now, let alone how they were going to handle a retroactive consent form, a retroactive payment. So if this goes in before the trial and it's not an amendment, this is not an issue. But if you put it in as an amendment, you got to think about that. What's going to be the trigger for consent? Is it randomization? Is it completion of treatments? Is it split? These are questions that we have to answer. Okay, these are the lessons that I just walked you through one by one. Hopefully that's been helpful. And with that, I will turn it over to our virtual speaker.
Thank you so much. A lot of that is close to my heart, and looking forward to the Q&A. As a reminder, also our virtual attendees, please feel free to put something in the chat. We do have someone monitoring that, and we will be happy to read your questions or comments. And let me go ahead and introduce our virtual attendee, presenter, Dr. Chabra. He is on the call, so he will be available during the Q&A. He's a professor of medicine at the Mayo Clinic, College of Medicine and Science. He has been practicing as a hematologist oncologist and stem cell transplant physician since 2012. He trained at the Cancer Institute of New Jersey and Stanford University. And his clinical interests include myeloma, transplantation, and cellular therapy. And he is a study co-chair for SWOG 1803 Dramatic trial, which is a multiple myeloma trial. So he has prepared this presentation. I'm going to go ahead and
Good afternoon. My name is Saurabh Chabra. I'm a professor of medicine at Mayo Clinic, Arizona, and my clinical research focus is multiple myeloma, transplant, and cellular therapy. I'd like to start by thanking the organizers of this symposium for giving me the opportunity to present. Today, I'll be talking to you about clinical trial design and how we can impact representation in trials by using a SWOG clinical trial as an example. Here are my disclosures. Let me begin by discussing the scope of the problem and provide some background data that gives us a magnitude of the problem at hand, which is disparities in cancer trial population. Nearly 20 years ago, Murthy and colleagues attempted to characterize the representation of racial and ethnic minorities, the elderly, and women in cancer trials sponsored by the National Cancer Institute from 2000 to 2002. The main outcome measure was enrollment fraction, which is defined as the number of trial enrollees divided by the estimated US cancer cases in each race and age subgroup. So these investigators showed that the cancer research participation varied significantly across racial groups. Blacks and Hispanics were significantly less likely than whites to enroll in NCI-sponsored cooperative group trials. And compared with a 1.8% enrollment fraction among white patients, significantly lower enrollment fractions were noted in Hispanics and Blacks at 1.3% each. In comparison to whites, after adjusting for age, cancer type, and sex, patients enrolled in 2000 to 2002 were 24% less likely to be Black. The investigators also assessed temporal changes in enrollment among racial groups, comparing patients enrolled in 2000 through 2002 with those enrolled in 1996 through 1998. And it was found that although the trial enrollment rate increased by almost 50% from 96 through 2002, the proportion of trial participants who were not white actually declined during this period. In a recent review of more than 112,000 patients recruited in 230 oncology trials that led to FDA approvals between 2008 and 2018, only 3.1% patients were Blacks and 6.1% patients were Hispanics. This represented no more than 22% and 44% of their expected representation if compared to their proportion of US cancer incidence, respectively. Black and Hispanic patients were consistently underrepresented compared with their expected proportion based on cancer incidence and mortality in the US. For all cancers together, the EID and EIR, which stands for Enrollment Incidence Disparity and Enrollment Incidence Ratio, were unfavorable for Blacks and Hispanics compared with whites and Asians. Similarly, the EMD and EMR, which stand for Enrollment Mortality Disparity and Enrollment Mortality Ratio, were unfavorable for Blacks and Hispanics compared with whites and Asians. The results were consistent for almost all cancer types, with underrepresentation of Black and Hispanic races compared with whites and Asians, although the degree of disparity varied across tumor types. The enrollment incidence disparity for myeloma was second highest among all cancers in Blacks and Hispanics, with an enrollment to incidence ratio of 21% for Blacks and 4% for Hispanics. The enrollment mortality disparity was equally high. Diversity in treatment clinical trials that reflects known incidence and burden of cancer in specific racial groups is critical for appropriate interpretation of the role of treatment specific to the patient group. Over the past decade, there have been several guidance documents, position statements, and recommendations from regulatory agencies and cancer societies to address disparities and underrepresentation of elderly and racial minority groups in cancer clinical trials. Success of these measures can only be achieved through partnerships between all stakeholders involved in the clinical trial journey. One of the areas that is considered key in improving diversity in clinical trial participation is clinical trial eligibility criteria, which define the study population and reduce patient risk. However, traditional criteria restricted populations to the healthiest patients, promoting disparities in enrollment. In 2017, ASCO, Friends of Cancer Research, and FDA issued guidance on modernized study criteria to improve the generalizability of results, to achieve greater efficiency of clinical trial conduct, and to provide increased opportunities for patient participation. Clinical trial eligibility criteria are designed to protect participant safety and to define an appropriate study population. Reiner and colleagues aim to determine the impact of eligibility criteria on disparities in pancreatic cancer clinical trial participants at Virginia Commonwealth University from 2010 to 2019. They showed that using traditional criteria, Black patients are more likely to be ineligible for participation compared with white patients due to hypoalbuminemia, HIV, hepatitis B, and hepatitis C. They were also more likely to be ineligible because of renal dysfunction, recent coronary stenting, and uncontrolled diabetes. So the investigators of this study showed that traditional eligibility criteria differentially excluded Black patients from participating in pancreatic cancer clinical trials. And this leads to a double hit phenomena where patients with pancreatic cancer from minority backgrounds are less healthy and are unlikely to be provided opportunities to participate in clinical trials. Narrow clinical trial eligibility criteria can compromise the relevance of results to the broader patient population with the disease. Exclusion of certain patient populations or disease characteristics is common in cancer clinical trials and leads to underrepresentation of older adults, sex, and racial minorities and patients with well-managed comorbidities. ASCO and Friends of Cancer Research, in collaboration with the FDA, modernized criteria with revised guidelines on organ dysfunction, prior or current malignancy, and comorbidities as potential strategies that could expand trial eligibility while maintaining patient safety. And these strategic eligibility criteria revisions adapted from ASCO and Friends guidelines could equalize ineligibility rates between Black and white patients. These revised criteria included removing historical, controllable, and manageable medical conditions. And it was shown that after applying these revised criteria, there was no longer any difference in ineligibility rates between Black and white patients. So through revised eligibility criteria, there is a potential for more equitable clinical trial participation and producing more generalizable results and reducing disparities in access to treatment. There are several potential benefits and risks of expanding eligibility criteria. The primary purpose of eligibility criteria is to protect the safety of the participant who may have characteristics that place them at increased risk for an adverse event from the intervention. Thus, the arguments against the use of broader eligibility criteria center on the concern that the development of an effective drug could be jeopardized if a serious adverse event occurs in a patient population that is inherently sicker or vulnerable. Inclusion of some patients may require additional screening or monitoring or engagement of additional expertise to manage safety issues specific to that patient population. This would help to mitigate risk in these patients, but could also increase trial cost and complexity. Although incorporation of an expanded trial population could present additional operative considerations, this practice could be accompanied by incentives such as potential for expanded label indications resulting in competitive marketing claims. In addition, there is a potential for inclusion of additional information in the label's prescribing information to help guide clinicians in adjusting administration and dosing in different populations. Adequate data generated in clinical trial on underrepresented populations, such as those with organ impairment, may obviate requirements for post-marketing studies. Laboratory tests that predict and assess toxicity are critical for determining whether a patient can safely enroll on a clinical trial. However, some laboratory reference ranges that are included as trial eligibility criteria are totally arbitrary with minimal justification for their use. Reference ranges or intervals that lack scientific rationale or differ from routine clinical care often result in biased clinical trial outcomes, as healthier, more homogeneous trial population may not represent the patients actually treated with a drug once it's approved. It may slow down clinical trial accrual as well. So ASCO working group tried to understand the practices related to laboratory eligibility criteria in an extensive review of more than 100 industry-sponsored trials, including 16 trials that only enroll patients with hematologic malignancy. And one of the lab tests that reflects marrow function is the absolute neutrophil count, or ANC threshold, which provides a margin of safety for myelosuppressive treatments. And this review showed that there were different ranges of eligibility criteria with regards to the ANC cutoff. For example, majority of the protocols, especially solid tumor trials, had an ANC requirement of more than 1,500. But an ANC of more than 1,000 was also a common requirement, especially in the heme malignancy trials. And a few protocols specified anywhere from more than 500 to more than 2,500. Let's change gears a bit. So with that background information, where are we with the currently active trials such as SWOG S1803 in terms of clinical trial eligibility and the opportunity to improve representativeness? Let me give you a little bit of background on the SWOG S1803. SWOG S1803, or the dramatic trial, is a phase 3 randomized trial investigating single agent lenalidomide versus lenalidomide plus subcutaneous daratumumab as maintenance therapy following autologous transplant. Adult newly diagnosed myeloma patients up to 75 years of age who are within 18 months of starting induction and are without progression at any time since diagnosis are eligible. There are 3 registration steps. Step 1, or enrollment, may be before or after autologous transplant. And if before, transplant should be completed within 18 months from initial or the step 1 registration. This trial has 2 randomizations, which are also referred to as step 2 and step 3 registrations. Patients are to undergo first randomization to lenalidomide or lenalidomide with daratumumab within 180 days from autologous transplant. MRD is assessed using next generation sequencing on the clonoSEQ platform prior to the start of maintenance and then annually. The randomized treatment will continue for 2 years, at which time patients who are MRD negative at less than 10 raised to the power minus 6 undergo second randomization or step 3 registration and either continue maintenance on their assigned arm or discontinue maintenance. The primary objective of this trial is to compare overall survival between lenalidomide plus daratumumab as the experimental arm and lenalidomide as the control arm. The objective of second randomization is to compare overall survival of MRD negative patients who continue maintenance on each arm versus those who discontinue. An early readout of the trial will be the 24 months MRD analysis as early as 24 months after all patients have been randomized. Here is a timeline of the important milestones for the trial. As you can see, the first patient was enrolled in August 2019. A total of 1,100 patients are to be accrued to step 1 to allow for 950 patients for first randomization. Once 1,100 patients were enrolled to step 1, the trial was temporarily close to accrual in the end of March. And this trial has surpassed the projections, as we can see here. And we are currently working on an amendment to revise the sample size. And so we expect the trial to reopen soon, and we'll have a sample size of 1,421 patients in step 1. And one of the reasons for this rapid accrual is that the trial was open to enrollment at more than 600 locations throughout the country, most of which were NCORE sites. However, if you look at the patient contribution to the trial, we can see more than 50% of the patients on the trial came from 15 academic sites, which I find very interesting. I mean to say, we need both academic sites and community sites for a successful completion of enrollment and in a timely manner for a large trial such as SWOG S1803. We cannot depend on only the academic sites or the community sites to move forward in clinical research. Here's the current enrollment status. As of last week, 1,141 patients are enrolled or step 1 registered, of whom 1,030 have undergone first randomizations. 330 patients have completed 24 months of maintenance therapy, and 90 patients have made it to second randomization at this point. Lenalidomide post-autologous transplant has been established as standard of care maintenance therapy based on several large phase 3 randomized trials. The major toxicity with lenalidomide is hematologic, with more than 40% patients experiencing grade 3 to 4 neutropenia and approximately 15% with grade 3 to 4 thrombocytopenia. And this has been seen in trials of lenalidomide maintenance as single agent and in combination with daratumumab in a frontline newly diagnosed myeloma treatment clinical trial for transplant ineligible patients, as shown here. And quick reference, grade 3 neutropenia is absolute neutrophil count less than 1,000 but more than 500 per microliter, and grade 4 neutropenia is absolute neutrophil count of less than 500 per microliter. Of note, the degree of hematologic toxicity was significantly less in patients receiving single agent lenalidomide as shown here. So in this trial, we required that patients had an adequate bone marrow function as evidenced by absolute neutrophil count of 1,000 and above within 28 days prior to first randomization, and therefore starting treatment on one of the two arms, which is quite logical and justifiable given the hematologic toxicity seen with both drugs, more so with lenalidomide, which was part of both the maintenance treatment arms. In addition, we had clear dose modification guidelines during maintenance therapy so as to manage the toxicity of neutropenia among other toxicities. In the setting of grade 3 and higher neutropenia, meaning absolute neutrophil count of less than 1,000, the protocol recommended holding treatment and resuming lenalidomide and or daratumumab depending on the attribution when the neutropenia resolved to grade 2 or less, meaning neutrophil count of 1,000 or higher, as we see in the two tables shown here. Lenalidomide dose was to be reduced by 1 level upon resuming treatment. However, a few of our site investigators noticed that their patients with Duffy null red cell phenotype, also known as benign ethnic neutropenia, had difficulties on the trial in that they required treatment interruptions for grade 3 and higher neutropenia more frequently than other patients, and then had to be restarted on a lower dose of lenalidomide, which was probably less likely to be effective. Some investigators were even holding treatment with daratumumab, which is less likely to cause neutropenia, but certainly is a possibility. This was shared on social media, such as these tweets, which are from the public domain. They're not confidential. And as study co-chairs, it is important to listen to what the site investigators are saying about the issues they're dealing with. So we explored this topic further. We know that individuals of certain ethnic minorities can present with persistently low ANC of less than 1,500 without significantly increased risk of infections. This condition has been referred to as benign ethnic neutropenia, which is correlated with homozygosity for a single nucleotide polymorphism in the promoter region of DARC gene, Duffy antigen receptor for chemokines. And that results in Duffy null red cell phenotype. The Duffy antigen acts as a cytokine sink that binds to inflammatory cytokines, so Duffy null individuals have fewer neutrophils in the periphery, but they have normal marrow cellularity and maturation, and they respond robustly to infection. Red cell phenotyping is the best way to identify benign ethnic neutropenia rather than race or ethnicity. And current evidence indicates that the risk of febrile neutropenia due to myelosuppressive therapy is not increased with Duffy null phenotype. Chemotherapy may be administered safely when the neutrophils range from 500 to 1,500, and prophylactic growth factors could be administered if chemotherapy is expected to lead to ANC of less than 500. Duffy null phenotype is common in individuals from sub-Saharan Africa in 80 to 100% cases, and Arabian Peninsula 50 to 70% cases, but is found in less than 1% of those with European or Asian ancestry. A recent study found that 67% of Black individuals have Duffy null phenotype, and there's a significant difference in ANC between Duffy null and Duffy non-null individuals, median of 2,800 versus 5,000 neutrophils per microliter. Additionally, 24% of individuals with Duffy null phenotype had an ANC less than 2,000 compared to no Duffy non-null individuals in this study. So the question follows, why is it important to account for Duffy null red cell phenotype in this trial? And that's because multiple myeloma disproportionately impacts Blacks in both incidence and outcome. For instance, a study published recently reviewing data from CDC from 2003 through 2016 showed multiple myeloma occurs twice as frequently in non-Hispanic Blacks compared to non-Hispanic whites. And so by not accounting for Duffy null phenotype, we would be contributing to a double hit scenario where patients who are in greater need are the ones who are deprived of the opportunity to participate in a clinical trial. So having a rigid, higher absolute neutrophil count reference range that does not reflect Duffy null phenotype can limit the access of certain patient populations to appropriate therapy and to cancer clinical trials. And the evidence supports that careful blood monitoring and prophylactic use of growth factor would provide sufficient safety for Duffy null individuals in clinical trials. And we can achieve that through explicit changes in eligibility and dose modification criteria. So we have used an ANC of more than 1,000 as the starting point for maintenance in all patients. And for Duffy null individuals, we will aim to maintain ANC more than 750 and modify lenalidomide and daratumumab dosing accordingly. So we would like to review the data from SWOG S1803 to see how many patients benefited from this minor modification to the neutrophil count threshold for dose modification. And I can anticipate the next step for us would be to incorporate a lower neutrophil count threshold for the Duffy null individuals in all the clinical trials. To conclude, I would say that changing the approach to eligibility criteria fundamentally requires a culture change across the entire clinical trials enterprise. In order to maximize the generalizability of trial results, eligibility criteria should strive for inclusiveness to enroll participants who are representative of the intended users of the intervention in the study. And we should do that taking into consideration the safety profile of the investigational therapy, standard of care treatment, and characteristics of the intended population. A standard of inclusion would give the investigators a responsibility to provide rationale and use their own clinical judgment as to why patients should be excluded from trial participation. Thank you for your time and your attention.
Sound for those virtual attendees. Keep trying. Okay, now they can. All right. Woo, okay. All right, so Mr. Moore is on the line, and again, he's an IRB professional and the Director of Institutional Review Board and Human Protections Program at the Wake Forest University School of Medicine, and we're hoping that he will provide additional insight on our recruitment, enrollment, and compensation or reimbursement of participants in the and how it affects and so forth. And then we have, and is Dana Sparks on the phone or? Okay, well, we're going to move forward, and I'd like to introduce Desiree, who came up. So Desiree Walker is an advocate serving on both the Recruitment and Retention Committee and DEI Monitoring Committee, and lives in New York, New York. So she is on East Coast time. Desiree has more than 20 years of experience as a health educator, trainer, and coach, with extensive service with SHARE and the Witness Project of Harlem. Both programs serve African-American communities in New York City. She also serves on the Patient Advisory and Community Advisory Boards at the Herbert Irving Comprehensive Cancer Center at Columbia University and the Tisch Cancer Institute at Mount Sinai. She serves as a research reviewer for the Conquer Cancer Foundation, the Cornell Center for Health Equity, Patient-Centered Outcomes Research Institute, and the Department of Defense Breast Cancer Research Program. She is president of the board of the Young Survival Coalition and a member of the NCI Central Institutional Review Board. And currently she is working on research projects with the sorry, my site is bad. Okay. Weill Cornell, Columbia University, Mailman School of Public Health, and the University of Wisconsin-Madison as a patient advocate and as a co-patient partner investigator on research projects with Memorial Sloan Kettering Cancer Center. She is a two-time breast cancer victor. So welcome panelists, and thank you so much for speakers for being here. And we would like to open up to any questions first. If anybody has questions from the audience about what you heard today or any comments, we would welcome you to come to the microphone. And I'm going to.
Hi, I'm Hildy Dillon, Vice Chair of the Patient Advocate Committee. So my first question is for Rick. I think I know the answer, but I think it's worth asking. And that is when SWOG is developing clinical trials, treatment trials, and there are several questions, translational medicine questions when we're looking for biomarkers. Very often there's not enough funding for that, and outside funding is sought either through a pharmaceutical company, but sometimes the Hope Foundation chips in. I know the Hope Foundation gave some money to get this going, but is there any possibility through Hope Foundation channels that, since we know now through your rigor and amazing experience, and I know not for lack of trying, did not get this approved. Is it just not approved, or is it just not approved because the NCTN doesn't have the capacity to fund something like this? So are they open if you get an outside source for funding?
I don't know. So even, okay, so if hopes to fund it, we still have to get it into an informed consent. And so it starts getting into NCI needs to approve it. Would Hope fund something? I think the answer is, I don't want to speak for Hope. All right. But my suspicion is, if it is something that pharma would not fund, so my example of a surgical trial, right? I think if we went to Hope and said, hey, we'd like to do something on a surgical trial. Now, there might not be a lot of transportation burden, but I'm trying to use an example where it was clearly not going to be pharma, right? Or if it was a some kind of de-escalation trial or something where pharma wasn't going to fund it, I think Hope would be amenable to it, but we still got to get it through an approval process. So we got it through the SWOG approval process, but I think you still got to get it through, and I'm not sure who we have to get it through at the NCI, because you're seeing DCP and CTEP, and we'll clarify this over the next several weeks, because I think we need to know the answer, not just at SWOG, but at the NCTN. If we're going to, we can't speak out of both sides of our mouth. We can't say we don't have enough information. We can't say we want to be equitable in terms of clinical trial participants and not acknowledge that transportation is a health-related social risk, and it is hurting us from a disparities in health equity point of view.
Thank you. And then I have a quick question for Dr. Chandra, and that is, if I did serve on the lymphoma committee. Our new advocate is here somewhere for over 10 years. And I'm really interested in your study, having been involved in blood cancer advocacy for a long time. If a patient is Duffy null and they are previously treated in the trial that you were presenting to us, how do you know if the ANC is below 1,000 from either the Duffy null, or is it actually from previous therapy or possibly a result of their disease?
Thank you for your question. I think that is a great question. And I think there is a little bit of judgment involved in answering that question. First of all, I would ask if the patient is currently on treatment. And if the answer is no, then I think it is likely that Duffy null is playing a big role. However, if there's a patient who has been heavily pre-treated in the past, then it is very hard to know. You know, if a patient who has had autologous transplant and multiple rounds of myelosuppressive chemotherapy, it will be very difficult to know. The problem is multiple myeloma patients, they don't stop treatment. So it would be difficult to anticipate a scenario where the patients won't be on treatment and you know they have low counts. I think most of the times we deal with issues where the patients are on treatment and we have to manage their treatment so that their neutrophils stay above a certain threshold. If the patient is Duffy null, I would probably have more confidence that I don't need to restrict their treatment, and I can you know, forge ahead. I can continue with the treatment rather than taking breaks from the treatment. If there's something that I get out of my experience with this trial and the literature search I had to do, I think it is that it is okay to continue the treatment and keep it over a minimum threshold, which is 500. You know, in clinical practice, I know in clinical trials, we are a little more conservative. And so we took a threshold of 750, but increase the use of growth factors and continue with the treatment is what I would do. But to answer your question in a simple yes or no, I think the answer is no. It's very difficult to know. And it's a judgment call most of the times.
Can I ask a question? My name is Jack Ayala. I'm also a patient advocate. I have a question about the Pragmatica trial. I think it was said that the primary reason for needing to do the phase 3 was that there weren't enough patients in the phase 2 to really feel confident and justify the 3-month improvement in overall survival. So if I have that right, does that mean that the eligibility criteria of the phase 3 trial, the dosages of the investigational drugs in the phase 3 trial were all the same as the original S1800?
So that's a great question. And so this is part of how we came to the pragmatic design. So yes, it's a robust trial, and but the confidence intervals are wider than in a standard phase 3 trial. So part of what we did to make this a pragmatic design, we do have much broader eligibility. So we don't have exclusions that we had in S1800A. We had lab exclusions. We had the standard exclusions that are in most trials. So what we did is, so this is a large randomized trial, so 700 patients. If we were doing this in a controlled setting, it'd probably be in the range of 300 plus or minus patients. So it's kind of double the size of the phase 3 trial we would do in a more controlled setting. So then the goal is that randomization washes away all sins, and that it all comes out and it's a large enough trial to see that difference. We also, in the statistics of this, have a lower bar. And so the hazard ratio, instead of 0.69, is closer to 0.75 to 0.8. And because we know that these are not the same types of patients and maybe less robust patients that were in the first trial.
And I want to, is this on? Yep. And I wanted to comment that we have Dana Sparks here. I was calling your name and I saw you come in. She is the Director of Operations and Protocols with SWOG and has been with SWOG operations for more than 30 years. And the last 12 as the Director of Operations and Protocols. So your educational background is in sociology of medicine, and I understand that you're going to give us some updates on the NCI and some of the efforts for decentralization.
Yeah, so simplification and flexibility in clinical trials has been something that the NCTN has always been interested in. I think that, things go in cycles. And so, most recently with a lot of the advances in translational medicine, we had gone in the direction of more and more complex studies, the platform trials, the biomarker eligibility for trials. But I think the strength of the NCI has always been in simple studies. And I also think that that's one of the reasons that we've been better than maybe our pharmaceutical company colleagues in registering diverse patients to our clinical trials is because the access is there through the institutions and the trial procedures are as simple as they can possibly be for the most part. We do have some very complex trial designs that have a lot of bells and whistles that require more from patients and make it less likely that diverse patient populations will participate, but those are the minority. The majority of NCI trials tend to be pretty straightforward and simple. And so one of the things that we learned through COVID is that there were a lot of requirements in place, and we learned that we could get past those barriers because we had to. And so there were things like being able to ship oral drugs directly to patients, being able to use remote consent, using telemedicine when possible. A lot of things we learned during that time immediately. It was immediately people started asking, why can't we do this all the time? There's nothing substantially different other than the fact that people don't want to be in the same place at the same time. There's nothing substantially different about the COVID situation than would be something that our patients would appreciate all of the time. So we started looking into that from the beginning, trying to figure out what are the ways that we can extend the flexibility to regular trial accrual and patient participation in our studies. So we started by outlining a list of all of those developments that had happened during COVID and been allowed during COVID, and then tried to look back and see what the regulatory underpinning for those were. Why was this allowed now when it wasn't before? What were the requirements? And it's some of the same questions we approached Pragmatic along with, is what are the assumptions about the regulatory requirements, and can we double check and make sure that those assumptions are real? Or is it something that we can get a variance from the FDA and say, you can do it in this case? And so we began asking those questions and pushing back on those abilities. And the more recent developments, we started calling it trial flexibility, but the FDA has turned that into what they're calling decentralized clinical trial design. It's kind of the same thing. There are bits of differences, but they're basically the same thing. And it's just ways of getting access to clinical trials out into the community and making clinical trial participation easier for individual patients. The FDA put out a guidance document last week, so we now have that. It's a draft guidance, so it hasn't been fully approved yet, but the direction that they took is very similar to what the NCI did. So it's reinforcing to some degree, and talking about things in terms of appropriateness. And so one of the issues that we've always had is who decides what's appropriate. You know, is it appropriate to allow remote consent for every trial? Or are there certain trial designs where it's more appropriate than others? And who makes that decision? So I had contacted the NCI at the end of last year and asked them, you know, kind of outlined all of these flexibility initiatives that we want to use. We want to decentralize as much as possible, and asked them what was possible through the NCI system to get consistent language and protocols across the board. And so they have now created a working group among all of the NCTN groups to talk through what those elements are, what's the right way to word them, and how to decide whether it's appropriate for each element to be used in individual protocols. So for example, if you have only IV drugs and the IV drugs are all investigational, there's not a whole lot of latitude that you can have to allow administration outside of a clinical trial setting. But if you have oral drugs, whether they're investigational or not, in certain stages of trial design, you know, if it's phase 2 or later, you probably have enough safety information that you're not going to be too worried about events that the patient is going to experience in taking an oral drug. So, you know, you may be able to have more latitude for shipping drug directly to patients as opposed to having it administered in an office. But the elements of the FDA guidance go into a lot of detail about what's appropriate, how can you allow a patient's regular healthcare provider to do some of the, to provide some of the elements of clinical trials data. And then there's a lot of discussion about what kind of agreement does the trial investigator need to have with the healthcare provider in order to make sure that they get the data. So there's a lot of things that still need to be worked out, but I think we're moving in the right direction. And the NCI has been pretty proactive about trying to pull all of this together. So I have some hope that, you know, maybe within the next year, we may end up in our protocols with a menu of flexibility initiatives that where we can like pick, you know, for this particular trial, because it's a phase 3 and it involves only oral agents, we can put the whole menu in. But for that trial, maybe we need to pick and choose and just put 3 or 4. So I think we're moving in the right direction. I'm happy to answer any questions.
Quick question, just a quick follow-up to Dr. Reckamp on the Pragmatica trial. I think I saw on the slide, and I should know this since I'm on the team, but I don't, so help me with that. I think I saw that targeted therapy candidates who meet other eligibility criteria might be eligible for it. So for example, would that mean that there are any patients with mutations like ALK or EGFR that could be eligible? I was just curious.
So potentially, yes, but they have to meet all the other criteria. So probably more likely somebody with KRAS or something that has gotten a KRAS inhibitor, because the caveat for things like EGFR, the non-smoking lung cancers, you have to have at least stable disease for 84 days. Immunotherapy has kind of minimal benefit in these patients. Many of these patients don't receive immunotherapy.
Right.
And so if they don't receive immunotherapy or have rapid progression, which we've seen in some, they wouldn't be eligible. And so what we're finding just from questions that are coming through is that most of those types of patients, the non-smoking oncogenic driven tumors, are likely not to be in the trial. But if they meet all the criteria and have had the appropriate therapies, something like ALK, I think, would rarely come in the trial because there's so many approved therapies.
Right.
And so they generally don't get into these types of trials.
Okay, I thought
Yeah.
so. I just wanted to make sure because there are a lot of those oncogene specific groups out there that might be interested.
Right. If they had response to prior immunotherapy, they could be and have had the appropriate treatments, they could be eligible.
Okay, great. Thanks so much.
And there is a comment in the chat that came in when Rick Banks was giving the hypothetical scenario of someone coming to a clinical trial numerous times and the financial costs. Naresh Kumar stated only 1 in 3 Americans can comfortably cover a $400 emergency expense based on recent survey data. So just talking about what is the actual, we're just talking about financial cost of being able to participate. I don't know if anyone wants to comment about that.
Yeah, I think the source for that is bankrate.com, if I'm not mistaken. And they do this regularly. And sometimes the amounts have changed. So this kind of threshold of $400, $500, or $1,000 is fairly common. And if you look at what we present in terms of transportation costs, it's pretty easy to hit that bar, right? And so if you think about the whole point is if you accumulate it, it's more real as to what the rather than it's only $55.25. No, it's really not. So.
I wanted to give Desiree an opportunity to comment on whether it be the pragmatic approach to trial design or patient reimbursement. What are your thoughts or considerations or feedback?
I guess before I even get to that, one of the things that I have heard over time is that those who are underrepresented in research, they've never been asked. And I think, before we start worrying about the financial and all the other barriers, we have to have people to ask the question. And I think that, one of the challenges, there was a slide that, talked about what's happening at the academic centers, but we also need to acknowledge that those who are underserved, as well as those that are underrepresented in research, many of them do not go to academic centers for a variety of reasons. That's not what we're here to talk about today. But I think it's important that we acknowledge that that's not where they are. And if the majority of the research is not happening in the community and academic centers are not necessarily working with the community, there again, that's going to create an issue. One of the things that I think is important, I have definitely brought it up on many occasions as it relates to financial toxicity and the barriers that those who have been diagnosed would have. No one really asks you how you're doing in any regard once you've been diagnosed. They're focused on treating the disease. It doesn't matter whether or not you can even afford, if you did oral therapy, whether or not you could even afford that. And so I think that it's really important that we really start to know who we are dealing with and that we really stop creating situations where we're not asking the right questions. And perhaps maybe we're not asking the right questions is because we feel that we don't even have the services to offer them if we do know what they're lacking. And so the sad reality is that financial toxicity and the need to be able to even get to appointments is real for many people. It's just that sometimes because of the state people are in and that they want to be able to have some hope, they don't even disclose the challenges that they're going to face in order to just get there, because this is their, perhaps in certain instances, a last opportunity in order to turn things around with their respective disease. And so I just really think that, part of the issue is that we're not really even knowing who we are really talking to. In order to try to make things more accessible at the end of the day.
Can I build on that? So it would be my hope that we can, if we differentiate between people who are not asked at a place that has a trial from people who are not asked because the trial is not close, it would be my hope that the transportation cost thing would help in that regard. I'm not referring a patient, I'm not suggesting to the patient that they should consider a clinical trial, which I don't offer, by the way, because the transportation cost would be too burdensome. So I'm hoping there's a little bit of room there for improvement on the not asked, but I'm specifically referring to the people who are not asked because they are not at the institution or an institution that offers the trial.
Okay, well.
Can I also follow up on
oh,
that? Because
Yes.
it's all about the ask. And I learned this at a recent lung cancer advocate-based conference. So it was a lot of survivors, a lot of patients that were there. And one of the speakers was an MD. I think he was at Dana-Farber or MD Anderson. I forget which, but the thing that he taught us that I was just so shocked. He asked a patient, or someone asked a patient of his if they would participate in a clinical trial. It was a radiation oncology-based trial where she would have to be there, every day for X number of days, back and forth, back and forth. And she said no. And so they said, oh, okay. And he heard this and he went back in and said, well, can I just ask you why you don't want to participate in this trial? She said, because my daughter has a baby and I have to babysit and I can't be gone. And so they figured out a way to reschedule the radiation therapy in a different manner so that she could in fact participate, maybe not on that trial, but at least get the therapy. She could have died if he wouldn't have gone back in that room and asked her the question. So, don't just ask why not if they say no.
Thank you.
Also follow up on what Desiree was saying. I'm wondering about the stuff Dana's talking about, which is really important about simplifying and making more flexible aspects of clinical trials. And I'm wondering how that connects with actually changing the people that were enrolling in trials. Like, does that really, do those simplifications or flexibilities really just make it more feasible for the clinics who are already connected to do the trials, which is really important, right? Because if they're not opening them, we can't do it. But it doesn't really, I don't know. I guess that's the question. Like, does it give the possibility for making other clinics or cancer centers or docs able to be involved in this enterprise?
I would just say that I think that flexibility is the key to so much, right? We have, and I would also go as far as to say that accommodating, because oftentimes people are not accommodated. This is the way we do it, and it's either you conform to this or not. And I just think that if we're going to say we're moving in a different direction, we really need to start to broaden our approach and our mindset and how we operate at the end of the day. The reality is that there may still be certain individuals with this flexibility that it still does not work for them. And I think that that's reasonable, but I think that we need to start to be more flexible, to realize that it's not always that we have to do things one way, because all groups are not the same and the needs are not the same, and they should be tailored to that need.
Can I make a comment here? Saurabh Chabra.
Yes, please go ahead.
So, I really appreciate what's been said today. But I'd like to make a point. It's one very important thing to get a patient on a trial, but for a maintenance treatment trial such as SWOG S1803, it's also very important that the patient stays on the trial, and that's where something like of a decentralized system or essentially telemedicine can come in very handy, because instead of having a patient come in every month for a face-to-face interaction, we could allow patients to be seen remotely via a virtual visit, a video visit, and as long as the patients are getting their labs on a monthly basis. But that'll take a lot of pressure away from the patients, and they could go on a vacation, and as long as they're doing well. And I think that their chances of staying on the trial will be much higher, and we'll be able to get all the data that we need for the trial. So I think that's one of the things that I'd like to see happen for SWOG S1803.
Thank you. We were going to give Brian Moore an opportunity to comment. He's virtual.
Yes. Thank you all. Can you hear me okay?
Yes.
Great. So really enjoyed hearing all of the perspectives and feedback from the presenters. I do have one additional comment, and it is in line with what Desiree mentioned, and that is regarding the informed consent process. And I say process intentionally because I think it's important that our researchers are communicating with our patients openly and being transparent through a discussion, not just displaying a 20+ page consent document that is left open for question, is rigid, and doesn't get to the importance of what is necessary for an individual to make the decision about whether or not they want to participate in a study. We all know that there are often trust issues, especially amongst certain demographics of people, but being open and candid and hearing the concerns and questions of our participants is very important and should be a part of the consent process, not just the consent document. Thank you.
Thank you. And I would comment on the informed consent process as well. I know at our site, we make every opportunity, if we have a patient that might actually be possible evaluated for a trial, we do approach every patient, and that's really important to our site. And I know that it takes engagement of the physicians and it takes engagement of the research staff to support that. It's not always convenient. And I will just comment on our treatment trials specifically. The physician is the one that provides the consent with the support of our research staff. And of course, the patient needs time to consider it. And it is a process, just like what Mr. Moore was saying, it's a process. And I always like to communicate to patients exactly that. If they have questions anytime, it's not, it's not a contract they're signing. This is something that we're want to continue to inform them. But one of the challenges also with treatment trials is the timing. If they're in clinic, they're not necessarily seen within the next few days, and patients have to make a separate trip just to sign the consent. And then we have to create a separate encounter. And those are some logistical things at our site that we find challenging, and we're seeing it more and more. So I'm just wondering if there's any opportunity, or if anyone would see a different opportunity for those challenges. I like the idea of being able to reimburse patients, especially if we're asking them to come back.
I do know that the CIRB has just recently updated and expanded their remote consent guidance. And so if your site has not been using the remote consent option, it's definitely something to look into. And the CIRB has guidance information on that. I often, it's kind of funny to me. I'll have sites who will call up and say, we have to bring in the patient to sign the consent, and then we have to bring them back in to register them. And I'm like, why do you have to bring them back in to register them? If you collected all the data for the registration, the patient doesn't have to be there. So that's actually one of the things I was talking about, is there's a lot of assumptions out there. And it's also, hate to say it, but it's a good fallback excuse to say, oh, the FDA won't allow us to this. The NCI won't allow us to do that. SWOG won't allow us to do this. And that's one of the reasons why we want to get that information into the protocol documents, so that sites can decide locally what's appropriate for that individual site and what practices can they adopt that make participation in clinical trials easier for their patients. The solution may not be the same at one site versus another. And so then the more we can put into the protocol documents that tell the sites you have this degree of latitude or that degree of latitude, then they can make their own decisions about what they want to use.
And I think we have a question first in the back, if you'd like to go ahead and.
Thank you. So I am really appreciative of the idea of simple trials and things. And what about the ancillary support services? So Dana, have you looked at how the auditors are going to be looking at this? Because for pragmatic, it said grade 3 and above, but a lot of our IRBs and others, they come in and they say, well, you didn't catch this grade 2. But we only have to report grade 3 and above. So have you talked about how the auditing will work?
So our auditors centrally are working around the actual protocol document. We actually have not looked into what auditors at the sites are looking at. So if they have their own internal audit procedures and they're built around the assumptions of this is an NCI trial, so you have to report grade 3s, then there may be issues with that. And especially, we have the documentation from the FDA, from the NCI, and from the CIRB, all that this design is acceptable. So if individual sites need that for their own internal audit processes, we can definitely provide that too.
And we haven't seen that yet, but we keep kind of refining and clarifying that piece in the protocol about what needs to be reported. And I think as we get more patients on, we'll get some education about how the sites are managing that and where they're having issues.
Yeah, I just wanted to address what you said about informed consent and struggling with asking the patients to come back to sign. I think that there's a real yin and yang to that because you want people to have time to process what's been explained to them. I was working with somebody recently who actually was asked to sign an informed consent for a trial before the clinician had actually confirmed her diagnosis. So, because everybody has a job and the person who was going to go in and talk to them about the trial and consenting to the trial didn't realize, this is like a checklist problem. Has anybody talked to the patient yet about their results? This was for a newly diagnosed patient, but it could happen for a patient who now it's confirmed that they're relapsed or refractory and there is a trial option for them. So I know what it is to have to come back for a second visit, but I think if you're going to do remote consent without having to first have a chance to have somebody sit down and explain that trial and why they feel this might be a good option for a patient, we're losing an opportunity there. So I think we have to be very thoughtful, as Desiree was saying, individual by individual, and case by case, because it's really important for the patients to understand what's going on and have the time to process and make decisions.
All right, thank you everyone for this great panel. I want to thank Dr. Recamp, Rick Bangs, Dr. Chabra, Kamara Mertz-Rivera, James Ryan Moore, Desiree Walker, and Dana Sparks for this great conversation. What I really take away from it is the question, the word for the day seems to be flexibility. What can we do while maintaining scientific integrity, but still really ask ourselves, are we doing this because this is the way things have always been done? We've always signed the consent in person. And what do we really need to actually be able to include people and really thinking about how flexible can we be with our procedures? I think especially when you consider now that we're kind of in this post-COVID time, how many things have we thought, you had to do in person that now we do virtually and we do okay. And we're still able to maintain those connections and still be able to do things. I think we should definitely try to dive into how much flexibility can we have, given that we have all these electronic means and all these virtual ways for people to participate, and how can we take off as much burden as we can from the patients and allow them to be able to participate, make it easy for them to say yes. So let's try to really think about what is important. See, what are the most important things that we have to have in a trial, but what are some things maybe procedurally that we could release so that people can, say yes, and to ask everyone and not make assumptions. And then when they say no, just check and see it. Why is it that they had to say no? Maybe there's something that we could do to modify things or adjust or accommodate to actually allow them to say yes. So we have these evaluations on the table. Please complete those and leave those so that we can address other topics in our future sessions, so that we can, continue this conversation on how we can make our participant populations most diverse. All right. Thank you, everyone. Have a good one.